Hi everyone, we are going to do macromolecules part 1 today, and there are 4 different macromolecules, and today we'll just be focusing on 2, carbohydrates and lipids. And I am trying to keep these recordings under an hour because that's how long a lecture should be. So I'm going to skip reading through all this so you guys can read through that on your own. One thing I want to review really quick though before we go on are the 2 terms. I'm just going to write them here. Hydrophobic and hydrophilic. So anytime you see hydro, think of water. And then if you have a phobia of something, you're afraid of it, so we call something that's hydrophobic water fearing. And something that's hydrophilic, the word philic means love, so water loving. And so talking about those hydrogen bonds from the last lecture on water, those weak bonds between the partial charged and polar ends of the water molecules, something that is water loving can be broken apart by the water, like the sodium chloride that we looked at, and something that is water fearing does not want to break apart and dissolve in water. And we'll look at a couple examples of this today. So I just wanted to get those definitions out there. And then if we look down here, our question of the day is, where do fruit smiles go when you eat them? So fruit smiles, I don't know why so specific, but these are like a great value brand. I don't want to say like fruit snacks. I think they're really sticky. They get stuck in my teeth. But anyway, where do fruit smiles go when you eat them besides into your stomach? We'll get into a little more detail than that. Today we're going to be talking a bit about digestion. So for the pre-lecture assignment, I'm not going to read through and fill that out either because that's something you should have done on your own before you come to watch the lecture. So make sure that you've read those pages and filled in the blanks below. But I do want to go over this a little bit to make sure you guys understand. So organic molecules or macromolecules are composed of subunits. And these are important terms to know. I would circle them, underline them, however you want to mark that they're important. But the words monomers and polymers. So mono means one, and poly means many. So with these macromolecules, the individual subunits are called monomers. And think of them as individual building blocks. So if you were to think of these molecules as like Legos, for example, a monomer would be like a single Lego block. When these monomers bond together, they make something called a polymer. And again, poly means many. And so, think of polymers as a string of monomers linked together. So a polymer would be like the castle, the Lego castle, or the final result of whatever you are building with the Legos. And they're just made up of a whole bunch of these little single Legos. So a macromolecule is basically just a really long polymer. And macro, we're doing a lot of like, I want to call it medical terminology or science terminology, but macro means big. So basically when we say macromolecules, we're talking about a big molecule that is a really long polymer made up of monomers. Alright, so if we look at this picture right here, there are two different types of reactions. There's a dehydration reaction and a hydrolysis reaction. So, I don't know if that's focusing on that. There we go. So this is a dehydration reaction, and think of dehydration as something that builds polymers, so it helps stick monomers together. And in this reaction, a water molecule is removed to bond the monomers together. So if you look here, we've got these subunits, which are like a whole bunch of monomers connected together. And if you look at the top, you have, and these are usually just hanging off the edge of some of these monomers, you have an OH group and a hydrogen. And when those come off, they'll come together to form water, so you'll lose a water molecule. That's why we call it dehydration reaction. Because if you think about it, if you lose too much water, you become dehydrated. And as that water molecule leaves, it allows these two monomers to bond together. So these dehydration reactions help us to build these big polymers or macromolecules. Because it frees up these bonds here so they can bond together. Alright, so let's make sure we have this filled in. How polymers or macromolecules are built and broken down. So the dehydration reaction builds polymers, and in this reaction, a water molecule is removed to bond monomers together. Like we see in the picture here, there goes water. It leaves spots where these monomers can bond together. So when you hear dehydration, again, think building, and water is removed. And on the other hand, we have a dehydration reaction, a hydrolysis reaction, which breaks apart polymers. And in this reaction, a water molecule is added to break monomers apart. So this is the same thing that we just looked at, except for it's going in reverse. So we've got some monomers here. This time we have, let's say, 4 monomers that are bonded together, and it'll lose a water molecule. It's coming off right here. And that allows these to break apart and become a smaller set of monomers. And this happens a lot in digestion. And the word lysis here, anytime you see the word lysis, that means to break apart. And then again, hydro means water, so using water to break apart polymers, and then a water molecule is added. So again, this happens a lot in digestion because it helps us break down these long polymers into smaller subunits. And here we should have the word monosaccharide. Mono, again, meaning one. And this is something that consists of only a single sugar molecule. So here we have an example of a monosaccharide. This is glucose that you see here. And it's the monosaccharide that our bodies use for an immediate source of energy. Alright, so it's all broken down to the smallest unit it can be, and so it's just a fast, immediate source of energy. And underline immediate. And a couple other examples are fructose and galactose. Down here, a disaccharide is made by joining two monosaccharides together with a dehydration reaction. Remember, dehydration is builds polymers. So if you stick two monosaccharides together using a dehydration reaction, you get a disaccharide. And some examples of this are lactose and sucrose. So if you look here at this image, you have two monosaccharides, two glucose molecules, and with a dehydration reaction, they can become bonded together to make a disaccharide. And then with a hydrolysis reaction, we can also break them apart. So I'm going to repeat the main things that you should know here from this page. Know that a monomer is like a single building block, and when you stick those monomers together with the dehydration reactions, you get polymers. Polymer meaning many. And then make sure you understand the dehydration and hydrolysis reactions and know that dehydration builds molecules or builds polymers, and we lose a water molecule during this. And with hydrolysis reactions, they break apart polymers, and a water molecule is added to break them apart. Alright, that was a lot of information on one little page there. Alright, so we're going to start off talking about carbohydrates. So these are polysaccharides, so they're long chains of sugars of glucose, usually glucose, so they're polymers, and they are immediate energy storage. So some examples are starch, which comes from plants. And it's usually found in the parts of the plants like the roots and the seeds. And then there's something called glycogen. And get familiar with this word because you're going to hear it over and over. But this is from animals. So glycogen, there's a picture of it here. Got all these monomers bonded together to form this polysaccharide. And glycogen is found and stored in the liver. And then a little bit in the muscles. And I'm going to circle these two bullet points because these are two things that you should know as well. that starch and glycogen are examples of carbohydrates, which gives you immediate energy storage, or intermediate, and starch is from plants, glycogen is from animals, and it's stored in the liver and a little bit in the muscles. So what happens is you eat a starch, and what's going to happen is the digestive system breaks it up, and remember the process that does this is called hydrolysis, to break apart. And it will turn it into glucose, a glucose monomer, which goes into the blood. Which then gets sent to the liver. And the liver builds it into glycogen. And this is done by, remember when we build it together, we call this a dehydration reaction. So an example of this digestive process, and you may have done this before, but digestion can actually begin in the mouth with your saliva, and there's an enzyme called amylase that specifically breaks down starches into glucose. And so if you have a saltine cracker and you put it in your mouth and let it get all gross and soggy in your saliva, you'll notice when you first put it in, it tastes like a saltine cracker. It tastes starchy. And after a few minutes, you'll notice that it starts to taste sweet. And that is that hydrolysis actually happening within your mouth because of that enzyme turning the starch into the glucose monomers, and then you can taste the sweetness. So that's kind of a cool thing to try if you have those on hand by any chance. And another part of the job of carbohydrates is to provide structure. So in plants, you find something called cellulose. And this makes up plant cell walls, which gives the plant cells support. And sometimes we call this fiber when you're referring to it in your diet. And it's indigestible. To most animals. That's why it's important to have a lot of fiber in your diet, because it helps things kind of move through your digestive system. An example of an animal that can break down cellulose is a termite, because they eat wood, and that's primarily cellulose, so they have to be able to break that down. But why can't we absorb that cellulose? It's because it requires a special enzyme to break it down, and humans don't have it. So again, we can eat it. We eat a lot of cellulose, but we can't break it down into monomers so we can absorb it into our body, and instead it's what comes out at the end. So back to the question of the day and the fruit smiles. Where do they go when you eat them? And we have the ingredients listed here. So if you look, the number one ingredient is sugar. We've got corn syrup, apple juice, modified food starch, whatever that is, and cornstarch. So basically a lot of sugars and carbohydrates. So where does it go when you eat them? Well, if you look at this little process that we've drawn up here, that's what happens. Where does it go? So you eat it, your digestive system starts to break it up through those hydrolysis reactions until it gets to glucose, which will either be used as an immediate source of energy, and any excess will go into the blood, to the liver, which will build it into this glycogen polysaccharide by the dehydration reaction process. So good old fruit smiles. All right, now we're going to move on to lipids. So lipids are molecules that share a common feature. And again, this is something to remember, so I'm going to circle the bullet point. They're hydrophobic. This is something that we talked about at the start. So they're water-fearing. They don't like to interact with water. And they are nonpolar. Remember, water is polar. These are hydrophobic and nonpolar, meaning they're uncharged. So lipids, this is why they say you can't mix oil and water, because the oil, which is a type of lipid, is hydrophobic and it doesn't want to mix or mingle with those water molecules. There are three main types of lipids with various functions. The first one is called triglycerides. And these are used for energy. There's a type of lipid called a phospholipid. And these will become very important when we talk about cells, but they make up cell membranes. Sorry, my writing's a little crooked there. And then the third are steroids. And when you hear that, I want you to think of hormones. So let's start with talking about triglycerides. So a triglyceride is a polymer. It's made up of a bunch of little monomers, and this is for long-term energy storage. It's made up of 3 fatty acid chains. Which is where the tri comes from. 3 fatty acid chains that are attached to a glycerol molecule. And again, this is a type of polymer, and these are the monomers, the little building blocks that make them up. And these, we just talked about sugars and carbohydrates. These can store 2.5 times more energy than sugar. So they're more efficient at this long-term energy storage. And here is a picture. So you've got a glycerol, and then you have those 3 fatty acids. And through a dehydration reaction, remember to build a molecule, they bond with the glycerol, and you end up with this triglyceride. And then in the process, you lose 3 water molecules to leave spots for them to bond right there. So there's where the 3 water molecules came from. And now you've got a triglyceride here. And we call these fatty acid tails hydrocarbons because if you look, they're mostly just made up of carbon and hydrogen. And they have no charge. You don't see any charges here, which is what makes it hydrophobic. It's not going to want to react or mix with the partial charges of water molecules. So now let's look at unsaturated fatty acids. And this comes from plant and fish fat. These are typically liquid at room temperature. And if you look here, unsaturated fats are not completely saturated with hydrogens. And there's usually a double bond there between the carbons that takes the place of where one of the hydrogens would have bonded. So it's unsaturated, it's not completely filled with hydrogens like you would see here in a saturated fat like butter. And the double bonds here in the fatty acids make a kink or a bend in the fatty acid tails. So let's write that down. Double carbon-to-carbon bonds in fatty acids. I'm going to abbreviate. Make a kink or bend in the molecule. So you can see here, we've got an unsaturated fatty acid. We've got this double carbon-to-carbon bond that's not completely saturated with hydrogens, and therefore you get these kinks or bends in the molecule, in the tails of the molecule. And that's why these are liquids at room temperature, because you can't pack them together very well. If you are trying to build a cabin, you're going to want nice flat pieces of wood that can stack on top of each other. You wouldn't want something like this that has bends in it. That's not going to make for a good solid cabin. Over here, we have the saturated fatty acid. Oh, also, since they can't pack together well to be a solid at room temperature, they also don't pack into your arteries like saturated fatty acids. So saturated fatty acids come from animal fat. and they are solid at room temperature. Think like butter. So over here, we've got our saturated fatty acid. There's no double bonds between the carbons, and they're completely saturated with all the hydrogens they can possibly bond with. And it gives you these nice flat layers that can be easily stacked. There's no kinks in the tails, and this makes it a solid at room temperature. So the fatty acid tails are not kinky. I should say have no kinks. Maybe bend is a better word for this. I don't know. Alright. So these are easily stackable, which means they can stack up and build up in your arteries, and that is not a good thing. That can cause heart disease and other types of problems. So next we have trans fats, like vegetable oil. So remember, initially plant oils are unsaturated, but if you artificially add hydrogens, it turns into what we call a trans fat. So normally it would look something like this. You've got these carbons, you've got a double carbon bond, you've got the hydrogens. It's not completely saturated with hydrogens because of this double bond here. And if you add the hydrogens, it's going to take this double bond away and remove the kink. And you're left with a trans fat. So why would they do this? It increases the shelf life of the product. So, some examples of this would be like shortening used for baking, which I've never used shortening, but I always remember seeing friends' parents use it and just pull out this disgusting big old jar of white stuff, which apparently you want to use to bake with, and I think you don't even keep it in the fridge. It just stays in the can, and it will last for a really long time before it goes bad. So it increases the shelf life. Anything that's going to be sitting on the shelves in the store for a long time, like frosting or those. I am not a store-bought cookie fan, but those likely have trans fats in them to help with their shelf life. When you make a cookie from at home with your butter, your saturated fatty acids, they don't necessarily go bad, but I think they stop tasting good after a couple days once they're not fresh. But those cookies on the shelf with the trans fats, those may be sitting there for months. So we always hear trans fats are bad. So what are the health implications of this? Well, it's going to act like a saturated fat. Which, remember, doesn't have any kinks in its tails like this, so it's easily stackable. So what's it going to do when it gets in your blood? It's going to clog your arteries, potentially causing something called atherosclerosis, which is where you have that buildup of fat and plaque inside your arteries. So we did answer the question of the day, But this one's a little modified. Where do excess fruit smiles go when you eat them? So let's say you down an entire box of fruit smiles. So where does the extra go? So the glucose, remember our carbohydrate monomer, for our fast energy gets used right away. And if you can't use all of it right away, it's stored in the liver as glycogen. And if you get too much of that, your body only has so much place to store all of this. So if you you use up all the glucose, the extra gets stored as glycogen in the liver, and you store up on that, then it's turned to fats. Alright, so remember, glucose is the immediate energy that your body wants to use first because it's a monomer, it's all ready to go, it's easy to use. Any excess is stored as glycogen in the liver. It makes the glycogen from that dehydration reaction. And if we run out of space for that, it's going to get turned into fats, which is the long-term energy storage. Alright, next let's talk about phospholipids. So it's got the word phospho in it, meaning that it contains phosphate. And I think these are really interesting little molecules. So they make up, again, the phospholipid bilayer. Which surrounds your cells to make the cell membrane that controls what comes in and out of the cell. We'll discuss this more when we get to cells. But if you look at this cute little guy here, you've got a hydrophilic polar head. So hydrophilic meaning water loving, so it's going to be attracted to water. And you've got this hydrophobic non-polar tail. And not that you need to know this, but if you look at this chain, it's got a kink in it, so we can assume there's a double bond in there somewhere. You can't even tell what I just drew. Here, let's make it nicer. Carbon to carbon double bond. And so what's going to happen with that hydrophilic phosphate head, water loving, and the fatty acid tail here is hydrophobic. And if you've just got all these phospholipids hanging out, what they're going to do is they're going to orient themselves like this. So this whole part here is a way zoomed in version of the cell membrane. This is inside the cell. This is outside the cell. And those hydrophobic nonpolar tails are going to want to get away from any water. And so they're going to orient themselves like this. And so you have the hydrophilic heads on the outside and those hydrophobic tails on the inside. And again, very important when it comes to our cells. Again, we'll cover this more later in our cells chapter. Okay, our last one here is steroids. So an example of a steroid is cholesterol. And these are the, I don't know if building block is the right word, but they're like, maybe the base of these different hormones. It also is a component of cell membranes. And what it does is it provides stability in the cell membranes. And then you have hormones, and we're familiar with the sex hormones. These are just signaling molecules. And testosterone is the male hormone, and estrogen is the female hormone. And if you look here, this is interesting because this is a cholesterol molecule here. And if you look at testosterone and estrogen, they have this same initial pattern. They just have a few different functional groups attached, a few different double bonds in certain places, but yeah, they're all made of cholesterol. Okay, and that's all we have for today. I did keep that one short, 41 minutes. And next time will be Macromolecules Part 2, where we talk about the other two, which are, let's see, we did carbohydrates, lipids, and then fatty acids and, not fatty acids, sorry, nucleic acids and proteins. So we'll see you then.